This is Bob, the average somatic cell, and Bob's doing his everyday things like dividing when he's supposed to and keeping his organism alive. But suddenly, radioactive mutant exposure changes him. He's now a mutant cell. Wait, he's dividing repeatedly and ignoring everything. Bob became a mutant supervillain thanks to mutations in his oncogenes and tumor suppressors. We're going to be looking at the initiation stage of cancer, and radiation is the main cause of Bob's mutations. Oncogenes are a family of mutated genes that encourage cell proliferation. Proto-oncogenes are the normal version of oncogenes, and mutations in a proto-oncogene is usually dominant, which means only one allele needs to change, and eventually it leads to constant cell division. One example can be deletion. When we have a segment of DNA here, some of the genes would prevent proto-oncogenes from going too wild. If you delete them, proto-oncogenes will be uncontrolled. Tumor suppressors are a normal set of genes that keep cell division under control, repair DNA, and tell the cell when it's time to die. A change during carcinogenesis can inactivate this gene, and these genes are recessive mutations. There are two types: gatekeepers, which control cell proliferation, and caretakers, which care about the accuracy of cell division, like DNA. There's so many tumor suppressors and oncogenes out there. So let's look at a hypothetical situation in Bob's case. In Bob's case, he has an RAS gene mutation. And in the real world, the RAS gene is very common amongst humans. RAS helps with cell signaling pathways, cell growth, and cell death. In normal Bob's case, his RAS gene depends on growth factors. If there are growth factors, the RAS gene will turn on. And if there are no growth factors, then the RAS gene will be turned off. In mutated Bob's case, the RAS gene will always be on no matter if there are growth factors or no growth factors, so the cell will be dividing even when it's not supposed to. Let's look at Bob's tumor suppressor mutation, and that's in the p53 protein. It's a checkpoint between stages G1 and S. If the cell has flawed DNA, the p53 protein will stop it from proliferating and either fix the DNA or kill it. Without this, the cell will continue to divide, even with the flawed DNA, and this trait will be passed on to daughter cells. And basically, you'll end up with a bunch of cells with flawed DNA. Oncogenes and tumor suppressor inactivations are usually both contribute to cancer. Cancer is caused by a chain of mutation and is a truly formidable enemy to mankind. But if we keep on trying, we'll eventually be able to defeat cancer once and for all.